Welcome to another video for biology. In this video we're going to be discussing useful genetic vocabulary. Okay, so here's a list of the vocabulary words that we're going to be discussing in this video. So let's go ahead and jump right into the first two, homozygous and heterozygous. So what we have here is a diagram of the basically a generation diagram showing how these traits get passed along from your parental F1 and F2 generation. We're going to look at the parental. When Gregor Mendel was first starting off his experiments, he started off with what's called a pure breed for the peas. So he decided to look at a single trait. And for this example, he was looking at the trait of the color. Okay, so he had the green color and the yellow color. And he used two homozygous peas. Okay, so this is an organism having a pair of identical alleles. So if I look at each side of the chromosome, it has identical alleles. So it's coding for the same color. So if I were to get two peas that were green and I cross-pollinated those, then I would just continue to get more green. Or if I only cross-pollinated yellow peas, homozygous yellow peas, then I would only get yellow peas throughout. But what he did is that he got two homozygous pairs and he cross-pollinated those two, right? So as you know, independent assortment tells us that which allele is chosen is a random occasion. Okay, it's a random event. That's the law of independent assortment. So it could have been either one of those alleles that got shared, but it just happened to be the one that it was. Now again, because these are homozygous, there really isn't that many options for each of those in terms of the color, because if you look at the green pea, it only has genetics for the green color. And if you look at the yellow, it only has a genetics for the yellow. Okay, so we have now a fertilized pea, and it has again the genetics for either yellow or green, right? But being that the yellow is the physical dominant trait, then the appearance of this pea is always going to be yellow. And this is what's called a heterozygous. Okay, this is a heterozygous or a hybrid. Okay, so a hybrid is basically a pea, in this case, a pea that has, even though it's only showing, by physical appearance, it only gives off the yellow color, allele-wise, it has the genetics for both green and yellow, right? That's heterozygous, that's a hybrid. Okay, all right, but it always shows as yellow. And of course, if you look at your F1 generation, your filial 1, all of them are yellow, but they're all also heterozygous. They all have the genetics for the green and the yellow. Now, this is where it gets interesting, because when you cross-pollinate the peas in the F1 generation, you end up with our green color coming back up, okay, our recessive. Now, in order for a recessive gene to express, you're going to have a homozygous mix, okay? So if you look at the bottom, look at our green. It's a homozygous green. Now, if you look on the far right, it's a homozygous yellow. But the two in the middle, even though their physical trait is yellow, they're still heterozygous. They're still hybrids because they have the genetics for both yellow and green. All right. So again, all recessive expressing genes must be homozygous. Okay. Otherwise, if it's a heterozygous mix, you're only going to have the dominant physical trait that expresses itself. All right. Well, that checks off three terms: homozygous, heterozygous, and the law of independent assortment. Okay, again, remember, independent assortment is how the alleles whichever one gets picked, it's a random event. Okay, so the next terms that we're going to take a look at are phenotype and genotype. Now, I hope you find that as I define and explain these terms, it's going to be a nice smooth transition, because we've kind of already been talking about these. We just haven't said it in the specific terms. So let's first take a look. Let's go back and look at our generation diagram here. And we're going to look at the two words genotype and phenotype. Let's take a look first at phenotype. When I say phenotype, I'm talking about the physical characteristics of an organism. Okay? Just think phenotype, pH, physical characteristics, starts with the pH also. All right, so looking at each one of these peas, Okay, when we discuss phenotype, we don't really have to talk about alleles. We're not talking about genetics per se in terms of the genes or the code. We're just looking at what the expression, the physical characteristic is. And starting from the left, we have yellow, and then our first two, or our two on the right are, I'm sorry, the one on the left was green, and the two on the right are yellow. Okay, and that's just, again, referring to its physical appearance. Okay, now if we're going to discuss why they have that physical appearance, well, now we can look at the genotype. Now we're looking at the actual allele makeup, the genetic makeup. Okay, so we can actually ignore the color, okay, or the actual physical characteristic, and because when we look at the physical characteristic, we can't necessarily tell what the genotype is, right? Because if I look at my two on the right, I have two yellow peas, but just by looking at the color of the peas, I don't know what the allele, the genetic makeup is, because the one on the far right is a heterozygous hybrid, and the one in the middle is a homozygous, right? Okay. So the genotype, again, is referring to its actual genetic makeup. Does it have the gene for yellow, or does it have the gene for green? Okay. Now, one of the things that you need to get used to as well as we continue on, when we discuss a dominant gene, like the color, we're going to refer to it with a capital letter, like capital Y for yellow, and which is the dominant gene. And the allele for the recessive gene, we're going to refer, or we're going to diagram that with a lowercase y, okay? So then the genetic makeup for the one on the far left, you have two recessive genes, which would be two lowercase y's. The one in the middle would be, okay, guessed right, two capital Y's. Now what about the one on the right? Okay, the one on the right, because it's a heterozygous, right, it's a hybrid, it has a capital Y and a lowercase y, all right? Now anytime you have a capital Y or you have a dominant gene, of course it's always going to express that dominant gene, which is in this case yellow. All right, so again, those are phenotypes and genotypes. Phenotypes referring to the physical, the visual look of an organism. Genotype being the genetic makeup or the allele makeup of that organism. Okay? All right, so that's the next two, phenotype, genotype. Now let's look at a test cross. This is also known as a Punnett Square. Okay, this was developed in the 1900s by Dr. Reginald Punnett, and he helped with this diagram. We're actually able to better quantify or look at the probability of which gene is going to express. Let me give you an example. So if we look at the parental generation of our diagram here, we have two homozygous organisms which have two capital Y's and two lowercase y's, right? So we have a homozygous recessive, because the green color is the recessive gene, and then we have a homozygous dominant, right? So if we breed these, what are going to be the results? Well, the Punnett square again helps us. So if I take a look at my green recessive, okay, my Y recessive, that particular organism only can offer two alleles. It can only offer two Ys. So I would put my Ys on, and I can put them on the right over here. It doesn't really make a difference if they're here or here, okay? But it can, again, it can only offer two lowercase y's. And likewise, my organism on the right can only offer two capital Y's. Now, what would happen when these alleles mix? Now, if you look at the law of probability, when you have two organisms with two different options each, okay, I don't know if you guys remember back in statistics or perhaps pre-algebra, when you discuss statistics, if you have two options in each, then you would just multiply those two numbers, 2 by 2. That would give you, there's 4 possible combinations, right? Okay, so my first combination would be, and you'll notice here that I'll have my capital letters as the first ones. It's just good practice to do that. I like to have my capital letters show up first, and I'll explain why in just a second. So when we go over to the far right, again, this contributes a capital Y, a dominant, gene, and this one contributes a recessive gene. And then as we move down to the next quadrant, it's the same. And it's the same for all four, right? Okay. And what is the phenotype expression for these? Well, if you notice, I'm going to have a capital Y in every single one of these. And anytime I have a dominant gene that's present, that's the one that's always going to be the phenotype. That's the one that's always going to be expressed, right? And that's exactly what we see here with our diagram, our generation diagram. Now, remember what happens here when we can mix any one of these two together and cross-pollinate them. Okay, so let's look at the alleles. So our alleles for each one is heterozygous. So we have a dominant and a recessive gene, a hybrid in each one of these, and that's all they can contribute. This one can only contribute a capital Y and a lowercase y. And this one, the same thing. So our first quadrant, you're going to have a homozygous dominant, and then in our second quadrant, we'll have a heterozygous, a hybrid. And then if you look at that one, oh, now we have a recessive, homozygous recessive, and then another hybrid. Okay, so then what are these going to look like? The physical characteristics, now that we have the genotypes in. Okay, anytime you have that dominant Y, it's always going to come out as a dominant, the phenotype that's going to come out is going to be the dominant one, right? And of course we have our two recessive, homozygous recessive, so that must show our green, and that's where our green comes out. So as you can see, the Punnett Square helps us to guess the probability of which phenotypes are going to be expressed based off of the genes that each of these organisms is contributing. Okay, and also this is called a monohybrid cross. Mono, one, meaning this Punnett Square is only dealing with one particular trait, and that is the color of the pea. Is it going to be a yellow pea or is it going to be a green pea? Okay, so the Punnett Square helps us to find the probability, which is phenotypically speaking, this is a 3-to-1 probability. This is a 3-to-1 ratio. That means there's going to be 3 yellow and 1 green. But now, if we look at the genotype ratio, we're going to have 1 homozygous to 2 heterozygous to 1 homozygous recessive. So that's 1 homozygous dominant to 2 hybrid, and then to 1 recessive, homozygous recessive. Okay, well, that does it for test cross or our Punnett Square, and we also were able to go on to monohybrid. Now, dihybrid crossing, dihybrid meaning 2 characteristics as opposed to monohybrid meaning 1. Dihybrid crossing, Punnett Square, is a little bit more involved, so I'm going to actually reserve this particular idea for Useful Genetic Vocabulary Part 2 video. So that will come a little bit later. Okay, that does it for this. Good luck in your studying.